Thank you, everyone. Our final speaker for the morning session is Litt from the University of Pennsylvania. And Brian will talk about crowdsourcing and IEEE databases.
Great. Tell me, so we're about 20 minutes over time. How long should I talk?
No, not 20 minutes. You can talk for 15 minutes.
For 15?
Yes, because the session is supposed to finish at 9:30, but I think we'll change.
Okay, so I'll speak quickly. What I'm going to talk to you about today is really an effort that I hope to enlist everybody's help in, and that's data sharing and a new way of collaborating amongst our group. And this group has really been one that's been at the forefront of this. The grant that I described, the platform I described, was conceived and pushed through by this group. There were really two efforts. One was led by Andreas Schultz-Panago for Epilepsy, and we've heard a lot about that today. And the one I'm going to tell you about is IEEG.org and where it's going. And the point I wanted to make is really data and community is what's going to make these leaps forward. And if you notice that this leap that Ben Brinkman talked to you about going from is seizure prediction possible to embodying a device to showing 83%, 0.83 under the ROC curve, is something that was made possible by this platform and by data sharing. So just a little bit of nostalgia. There's the picture from the first meeting in Bonn in 2001, and I'll show you a little wet behind the ears. Dr. Greg Warrell here. Klaus and I were mere children at the time when we put it together. Jean Gottman has looked the same since he was 12, I think. And lots of other people, Rosanna Steller, who is now at NeuroPace, and a lot of people who, many were off hiking. Steve Schiff, he had hair, notice.
That was a long time ago.
So our lab, I'm not going to talk about our research today. We have some students who have talked about it, but I'll just say we're typical of many of the large labs that are here. We work in a variety of areas. We make sensors, flexible electronics, high-resolution sensors. We work in different kinds of brain devices in collaboration. Here's an example of, and the theme behind these is they all generate data that we need to collaborate on. This is a piece of work we published just a few months ago. This is graphene electronics over a hippocampal slice, low magnesium epileptic animal. There's the electrode recording this field potential, and these are calcium signals from the individual neurons that are labeled here, showing you some seizure-like bursts. If you look down here, in the filtered version, there's a spike, there's a heartbeat, this is a seizure-like burst, and just showing that this is the kind of data that are being generated. This is the kind of work that the Human Brain Initiative in the States is trying to push forward, where we try to get to the mechanisms of diseases, conditions like epilepsy, from the single cell level up to behavior. The issue is that they generate data. How are we going to be able to tell what works, what doesn't? The issue we all deal with is we have diverse data streams that aren't integrated. We have multidisciplinary collaborations. Just in this room, in many of the labs, we have engineers, physicists, physicians, neurophysiologists, different interests, distributed teams. We just heard about a team that was distributed across the entire planet. And your Australian accent is very interesting, by the way, I wanted to mention. No, just another example of international collaboration, right? We have a need, a lot of us here are pushing for translational work. We want to go from the basic science laboratory to animal models, to pilot trials, to pivotal trials, to clinical trials. Why is it that the data are all managed in different formats by different systems? Why can't you just initiate your trial, keep track of your data, and then when it's time to submit it to the FDA, just hit the share button with the FDA so that they can see all of it and all of those stages? And it turns out that there's a way to do that. We need a common data language between academia, government, industry, and the clinical world, and we need to change the incentives. Rather than holding our data close to our chests because we compete for grants and notoriety with publications, we need a system where people get credit for sharing their data. If we were in astrophysics or particle physics, or we were in genetics, we would have to share our data, and people would credit the data authors for collecting that information, and we need to change that. All of this introduces noise that slows down science. So, what do we need to collaborate? We need a central place to put data, where investigators have access control. If you put data to a place where you can share, it doesn't mean you have to. You have to be able to control who has access when. You need to be able to share easily across continents. Large data sets, like some of the ones I just showed you, they need to be able, in a place where people can get to them, and the cloud is a natural selection point for that. Although, many of us have invested in large data archives, so whatever the solution is has to federate. It has to point to your data archive too. You can't expect people to just, Greg Worrell, to take everything he has and just shove it up to the cloud tomorrow. We need to have processing power. Wherever the data are, they need to be able to be processed, and the cloud, again, affords solutions where you can create 1,000 instances of your processing machines to take on big tasks. We need to version our code and share it, so that if somebody publishes an algorithm that says does something, show it to me. Let me run it. Let me run it on your data and mine. There needs to be validation and transparency. And that's one of the key things about the Kaggle competition, right? Each of the people who participated posted their code, agreed to share their code so that it would benefit all of us. Then we need quality and security. So I'm going to do something really pretty high risk here. I'm actually going to go online to IEEG right now and just give you an idea of what this group has created. If it fails, I'll just turn it off, but it's pretty robust lately. Okay, first thing. Here it is. When you log in or go to the login page, that's what you see. There are 919 registered users as of right now that are sharing data sets, about 2,000 of them. Overall, these are people that keep their data private for individual collaborations, and some of them are made public, and you only have permission to see what you're given permission to see. Now, I asked one of the administrators to log on as an administrator. I don't have administrative privileges. I'm not going to open these databases. Everything that's done with the data is tracked, all the provenance. Dr. Morell can look and see who's opened the NeuroPace files from the group that's supposed to be sharing and collaborating on those. Dr. Cook, Dr. O'Brien can open and take a look and see who's working on their data and know what's going on. So there are 819 human data sets that are up here right now. And let me show you a little bit about where they're from. So a lot from the University of Pennsylvania. These are all intracranial data sets. From the Mayo Clinic, there are a bunch. More Mayo, more Penn. Children's Hospital of Philadelphia. FebStat, this is scalp data from febrile status. UCLA's got data up here. All collaborating with themselves. Mass General's got a big hunk. This is one of Sid Cash's mentees. We've got a group here collaborating through the DARPA RAM project, 8 centers collecting intracranial EEG, dragging and dropping it, having it go up to the cloud, all from different machines. It's put into the same format. All the images are co-registered the same way. All the cognitive tasks are done the same way, and the data is put in one place. Sound like something we could do? Absolutely. And it goes on and on. There are other groups. This is from DARPA RAM, CHOP, Spike. I don't know if I passed by it. There's NeuroPace data up here in a collaborative group, sleep studies and all that. Okay, great. So that's the kind of stuff that's there. I'm getting rid of the administrator. Actually, I'm afraid to do anything. Let me go on to the other place. So this is me. When I log on, I have an identity here. I can put together a collaborative research group, choose whoever I want, and they can share data. And everything that I do is tracked here. I opened this snapshot from NeuroVista, and this is, by the way, a map of where people are around the world. There's a hotbed of seizure collaboration here in Australia, but we have them in Africa, South Africa, Europe, the US, all over the place. Let's take a look at a couple of data sets quickly. Actually, I hope the resolution doesn't kill me on this. So I'm allowed to see 234 human data sets. I have 13 active projects that I've been invited to. So let me just search for data for a second. Greg Worrell has shared microelectrode human data that's up here. So let's search for it. And we have 6 human data sets. Some of these, Greg, if I'm not mistaken, are patients that don't have epilepsy that have recordings. Is that true?
Yeah, there's a group of normal, they have face pain, so their motor cortex
Right.
reporting prior to.
So take a look what we have. So I click on this study, and I look what's underneath it. I've got documents. In this case, there's the report. I'm going to open that. Actually, view docs. And this is the patient report with all the information redacted. There are the micros. This is the electrode implant. Here's all the patient details, anything you want to know about that patient. I can open the images. They're all downloadable, co-registered images. I can open the time series data, either a channel at a time, it's recorded at 500 hertz, or I can record the whole thing. Now, of interest, actually, there is a measure that's been run on this for data quality, DCN, data, I've forgotten what it stands for all of a sudden, but it shows you whether there are gaps in the data and where there are artifacts. So you can use that as a map if you only want to look at high accuracy data or where there isn't a lot of artifact. I'm going to open up the EEG. This has a bunch of channels on it with all its warts. Greg's not afraid to show his dirty laundry here. Are you, Greg?
There should be some good data in there somewhere.
No, there is. So if I take a look here, I can just move the amplitude, I can scroll, I can annotate, and my annotations show up here. I'm going to say lit, neat, blue, and put it on there, and there it is, lit, my annotation where the start and stop is. I'm going to remove that so Greg doesn't get mad at me. So that's an example of a study. And you can manipulate it, you can download it, any of you can work on it. Actually, you know, I'm on such short time that let me just show you some stuff and we can answer questions after if there is time. So let me show you another one. I'm going to search in the animal data. There are about 400 animal sets, and I'm going to go to dogs. Ben Brinkman said the dog data were available. So I searched. There are 9 dog data sets available. Here's one that's 451 days long. They're documents. Let's go to the time series. View the EEG data, and these are seizure marked. So there's a seizure on this dog. If I want to look at it, you know, in a longer duration, a longer time window, I can alter this window because the projectors change that. That's a seizure from day 7. This is a continuous data set. There's a seizure from day 406. It's all there if you want to collaborate on it. What about other things? Let's look at some animal data. I'm going to open another one of my projects. This is a project where we were asked to look at data from Aslepicanin, and there were some interesting things about this data and some controversies. I'm going to open the project, and I'm going to look for this data set. Oh, by the way, for each project, there are tools that you can associate where people upload code. There are people who have administrative responsibilities, and there are people who are allowed to be on the project team. Okay, so I'm going to view the EEG data. And anybody can annotate with an algorithm or by hand. I'm just going to pull all these off. And I'm going to show you, here's one. We wrote an algorithm to detect bursts because there was a big debate in these animals about whether this was epilepsy or not. So there's a burst. And I'm just adjusting the artifact with my mouse. The amplitude. 2 channels, and these are bursts. If I want to look at another one, another layer. These are a question of where are the spikes humming? Pardon me? Oh, right. There we go. So this is a spike detector that was a work in progress, and there's spike labels. These are done by the algorithm. We had a workshop here where we showed people how to do it. There are toolboxes. You can interact with the data, pull it down. We'll have a Python one very soon. We have it in MATLAB. We're working on R. You can do your detections and upload the annotations, and anybody can see them or nobody if you don't permit them. And so we can go through here and take a look at these. So do you get the idea? Very collaborative. People just need to give you permission. Let me show you one other thing to just entice you for a minute. And there's this thing here called the Human Neurovista data. So this is the 15 patients from our colleagues in Australia. I have permission to open it. This is not an open data set. The investigators are just starting to publish on this, but they're all interested at some point in the future in more collaborations, and they'll decide what they do with their data. But take a look. I'm just going to open a patient randomly. So this is a data set. There are different layers. They're the clinically confirmed seizures and the electrographic ones. I'm just going to look at the clinically confirmed ones. So this is the data set. This is day 5. I'm just looking at the seizures now. This is a seizure from day 421. This is a seizure from day 554 in continuous intracranial human data. It's all there. The documentation will be there, etc. All right, so what's the opportunity here? So we've got this set up. There's going to be a release of this data facility in October that's going to be pretty souped up. You don't have to enter data in fields like name and animal number and things like that. You just drag and drop your files, code, time series, imaging, documents, and crawlers go through the headers of these files and the content and build associations. And then you can do a regular text search like Google to say left temporal, Freiberg, right-handed, cognitive studies, and it will crawl through and give you a list. And the algorithms will learn over time. They'll give you a ranked list. You'll give it a thumbs up or thumbs down, and they learn. And this is running now, but it's not released. You have a presence here. You log in. You develop your project team. There's the box that you drag and drop your data into. And again, you can point to it on your own cluster. You can view and analyze data and run algorithms, either through the toolboxes on your own machines or parse to the cloud. So where are we right now? It is self-supporting. The grant for this is ending, but we have people writing us into projects, and the project is self-supporting. But it's not good enough for us to be run by graduate students, even though we have great graduate students. We want to make this a Red Hat-like utility. We're spinning off an entity at the NIH's request called Blackfin. And that will be, IEG.org will still stay open source. All the code for IEG.org is in JavaScript. It's all open source. You can get to it, you can hack on it, you can do stuff with it. But the idea is we want a professional level of support, a utility for each person to manage the data in their lab and then use it to share with other people. Sort of like you buy MATLAB or other licenses. We have device companies that are using it within their own area to share among their investigators. We have NIH-funded projects. I showed you the DARPA project. And the idea is to make this a utility that we can use. Here's just an example from that data set from ASLA Pitcairn. And the question was, are these seizures? Are those seizures? Are these? I think we would agree these are probably artifact. These are epileptiform. It's not clear what these are. In the strain of animal they were using, it turns out that those bursts were equally found in the sham animals and found in the head trauma animals who developed epilepsy and found in the head trauma animals who didn't. And it turns out that these animals probably have a native absence. They have seizures, they're control animals. So this is an issue. There are lots of strains that do that. These epileptiform bursts were found in the sham animals, in the head trauma animals without seizures, and in the head trauma animals that develop seizures. What we've noticed, we've now analyzed data sets from about 5 different labs with intracranial electrodes from different animal models of epilepsy. And guess what? The intracranial electrodes cause epileptiform discharges in the control animals. That's not surprising. We found that there's a lot of variability in the animals. Yes, these bursts, an increasing length of them, may be a biomarker for epileptogenesis, but there's tremendous variability. Some animals don't have them and some of them do. This study is not clinically significant, but it's interesting. The point of this is not to show you this, but actually what I wanted to show was this, that what you can do is make a pipeline like Homeng Ung, one of the graduate students, did. This is written in HTML, and it goes to the portal. So here's the contents of the pipeline. It calculates artifacts or the data quality, spikes, bursts, seizures. It establishes a session. Here's the MATLAB code. It's coupled to the portal. You can run it on IEG.org. And here's the output of the pipeline. These are animals from Finland, spikes, bursts, seizures, delta power, theta power, alpha power, beta power. You name it. You can click on these and get the plots. This is all of these animals run in batch from the portal from Finland. Here are preliminary results from head trauma animals. Here's UCLA data sets with these same things, spikes, seizures. Do you get the idea? When the data from all of these different labs is put in the same format and access is given to you, then you can do some pretty amazing things for collaboration. So, sorry, I know I'm out of time. This kind of collaboration and data sharing is getting tremendous play at the NIH. We've talked recently to Phil Bourne. There's now a chief data scientist that sits under the director of the NIH, and the heads of the NIMH and NINDS, the Neuroscience and Mental Health Institutes. They're interested in this. This was a blog that was written by Francis Collins, the head of the NIH, about the Kaggle competition. So, we're proposing, this is going to come online in October, use the portal, learn how to use it in the toolboxes, and let's think as a community on how to put something forward that sets us apart, that lets us do the kind of studies the cardiologists have done to really push this field together. Two last things. Data impact factor. Every data set on the IEG.org and on Blackfin has a unique identifier that ties it to the investigator, to the institution. And so when people publish papers, if you're using IEG.org,